Oh, okay. Good afternoon, everybody. Welcome to the CAES seminar series. I'm Joe Pignatello. I'm the head of the Department of Environmental Sciences, and I have the pleasure today of introducing our speaker for today's talk, Doug Brackney, who is an associate scientist in the Department of Environmental Sciences. He's been here for the past 6 years, ever since obtaining his degree from Colorado State University. His research is on vector-borne diseases, particularly those of mosquito and tick-borne pathogens that are of importance in Connecticut and the Northeast US. His interests in that field are in the areas of physiology, ecology, population biology, disease emergence mechanisms, vector host pathogen interactions, and pathogen transmission. And he's gone a long way toward establishing a strong reputation in these areas. Recently, he took a side path from his usual efforts on vector-borne diseases. In late March, after the experiment station shut down, he announced that he was going to work with collaborators at Yale to develop some new PCR-based assays and surveillance strategies for SARS-CoV-2, the virus that causes COVID-19. And so today he'll report on some of these results, which I think are very exciting and actually have already attracted a lot of attention. But I'll let him talk about that if he wants to. So Doug, go ahead.
All right, thanks, Joe. You kind of stole a lot of my first introductory slide
Sorry
talking
about
points.
that.
No, I mean, just to kind of reiterate, like, you know, when the pandemic hit, you know, obviously I've been working on vector-borne diseases as a virologist for many years, and when the ag station shut down, was working at home as many other scientists were, and research was really limited to research that could be done on coronavirus. And sitting there thinking, well, I'm a virologist, I've got the training, I've got the expertise. It would be great if I could actually apply my skills to, you know, help the effort to combat coronavirus, the SARS-CoV-2. And so that's when I did reach out to some collaborators at Yale in order to start seeing what I could do to assist in any way possible the efforts to combat SARS coronavirus. And so that's is what I'm going to be talking about today, is really sort of two projects. One is going to be focusing on how we were, how an approach we made to look at community level transmission, or I guess how widespread the pandemic was within our community, and then also how to improve diagnostic tests at an individual level and how that translates to a community level. So just a little background, SARS-CoV-2 is a severe acute respiratory syndrome coronavirus 2, and is the causative agent of COVID-19. It is in the genus beta coronaviruses within the family Coronaviridae, and the Coronaviridae get their name for the Latin term corona, which means crown, and that's sort of the appearance you get when you look at it through transmission electron microscopy and you actually see the virus particles. They have these predominant spike proteins that are protruding from the envelope that really give it this crown type of appearance. It has a positive sense single-stranded RNA genome, about 30,000 nucleotides. So that makes it actually one of the longest single-stranded RNA genomes for any of the viruses that have been characterized to date. And by positive sense, it means that when the virus first gets into the cell, it can actually immediately start translating and producing proteins. So it's going to start producing the proteins that it can use to replicate right as soon as the RNA gets into the host. And so that's why theoretically the RNA is infectious. So in the laboratory, you can actually condition cells to be able to get the RNA into the cells and actually produce virus from the RNA. Now, granted, in clinical settings or in laboratory settings, RNA is probably not dangerous to people working on it because the cells have to be preconditioned in order to be able to actually uptake the RNA before it is degraded. So there's very low likelihood that it's actually going to be infectious in the laboratory. Over on the right side of the screen over here, we could sort of have the phylogenetic tree breakdown of the Coronaviridae and going into the subfamily Coronavirinae and the different genuses. And then within the beta coronavirus, which is represented sort of by the left side of this this figure over here, this dendrogram is where you have these coronaviruses. In the last 20 years, there has been four epidemic/pandemic strains of coronaviruses that have emerged. The first one was in the 2002-2003, which is SARS coronavirus, the original SARS, severe acute respiratory syndrome. Then we also had an outbreak of a coronavirus in swine, so in pigs. And then we had emerging from the Middle East in the 2012-2013 is MERS coronavirus, and we still are getting cases popping up here or there, but luckily this is not, it's highly pathogenic, but at the same time it's not transmitted very efficiently between individuals. And then, and this, obviously the latest pandemic here, which started in 2019 out of Wuhan, China, is SARS is closely related to the original SARS, and so it's a beta coronavirus within the B clade here. So many of you are probably familiar with a lot of the disease. It's been all over the news, what to expect. There's a couple things I would like to point out, and that's really the duration and the percentage of people that show symptoms to when they're infected with SARS coronavirus 2. And you can see that a number of these symptoms, a high percentage of symptomatic people have, fatigue, cough, headaches, so there's a percentage along the top, 0, 20, 40, 60, 80. That's the percentage of individuals that have symptoms that report having these specific symptoms. And you could see that even after 14 to 21 days later, a number of these symptoms are still present in a number of the individuals for a long period of time. And this figure on the right really highlights that some people actually experience symptoms associated with COVID-19 out to 38 days, at least from the reports that were present or available at the time of this. So there are a number of symptoms, and they can be long-lasting even after you're done, battling the disease and are not shedding virus anymore. The demographics that we're seeing on the first figure over here on the left, we have case fatality rate on the y-axis and age groups on the bottom. Obviously, and this is pretty well known, that the majority of the cases that result in fatality are highly skewed to the older age group. So really over the age of 60, maybe 50, you could say, represent the bulk of cases that are associated with death. And within that, if you break that down further, there's also a skew towards the number of between male and female. So a higher percentage of those that do succumb to infection are male compared to female. And then why that is not 100% known, and there's active research groups looking into what the cause of this might be. When you look at the demographics of coronaviruses, and if you're looking at cases or deaths, we definitely see that minorities and specifically African American communities are being adversely affected by this outbreak. And this color coding basically shows if you're in the sort of the yellowish and orangish reddish area, that's showing that there's a disproportionately higher rate of African Americans who are affected either by deaths or by the number of cases in the states that are then associated with that. So this is really a disease affecting a lot of communities, a lot of urban communities, and a lot of there is a socioeconomic component to what to the spread, the transmission, and disease of this. So sort of the timeline is, and On December 31st, 2019, officials in Wuhan reported dozens of cases of pneumonia of unknown cause. And on the 7th, they actually identified it as a novel coronavirus on January 7th. By January 13th, it was found in Thailand. And then the first US case was actually identified in the US on January 21st of this year. Now, a lot of retrospective studies going back and looking at clinical specimens from before this are suggesting that the virus actually was introduced into the United States and circulating in China earlier, even between 1 to 3 months earlier than what actually these official dates represent. So the virus is there spreading probably undetected or being misidentified as other forms of disease such as influenza early on, and then once the virus got identified and it really became known is when we started actually being able to test people and determine it that if coronavirus was actually the cause of the disease. Over here on the right is sort of just showing how it spread across the United States with the darker colors representing increased transmission and number of diagnosed cases by state. And so many of you know those initial cases in Washington. Then we started getting identifying cases along the West Coast. It got into Chicago and up into Wisconsin, and eventually made its way out to the East Coast in March. And that's when we in New York, Connecticut, New Jersey really got slammed and became sort of the epicenter of the epidemic. This was in early March, and then as the pandemic continued throughout the United States, it's only increased in a number of other states. And just in the last month, Arizona, Texas, Florida, a number of other states were spiking. They're starting to come down now, which is encouraging, but there was it's sort of gradually swept across the nation into different communities and really kind of moved out of urban environments that were hit really hard and is starting on to permeate into sort of the rural communities as well. So early on, there was a big problem with being able to understand how widespread the pandemic was in the United States. And a lot of this had to do with the lack of being able to get tests. So there were people who were sick across the United States who were being denied coronavirus tests. They didn't meet the very strict criteria that were set forth by the CDC at the time as to who could get tested. And so these roadblocks really made it hard to kind of get an idea of what was going on. So, the question that we had was, how widespread is the pandemic at the community level? And so the next question is, well, how do we address that if we don't have tests? If we don't have the access to all these diagnostic labs and people are not being tested, how do we answer this question? So what we did is we said, okay, well, viruses, well, we know there's over 100 enteric virus species that can be found in contaminated water. So if you go to sewage plants, if you take wastewater, you can identify a lot of these viruses that are commonly circulating within our communities, from hepatitis to Norwalk virus. We don't really have a lot of polio virus anymore here in the States, but that can commonly be found in the sewage systems of places that are where polio is still available. And one thing you'll notice is you see in red there, I've highlighted coronavirus, and that represents actually other coronaviruses not associated with the pandemic. So we do have, there are a number of other strains. There's 3 or 4 other strains of coronavirus that routinely circulate throughout our communities that cause the common cold. And these viruses, we do know, actually can be detected in wastewater or in contaminated water. And so when a report came out suggesting that you could actually, so there's a study that was looking at different samples from individuals and where the virus could be found. It was found that it could be in people's feces. So this gave us an indication that potentially coronavirus could be detected in the wastewater, such as some of these other viruses. So there was a team assembled, which was led by Saad Omer and Jordan Pecchia at Yale. And And then we had a number of other people here working on this study. Ed Kaplan and Dan Weinberger did a lot of the modeling associated with this study. And Albert Coe really was also a driving force in getting this project going. There was a number of other graduate students, med students who assisted in the development of this, especially Alessandro, Dennis Wang, and Mike Wang, who are all working with Jordan Pecchia in his lab in environmental engineering. So what did we do? Well, back in March, Jordan had the great idea of reaching out to the Greater New Haven Water Pollution Control Authority to be able to start actually getting samples from them. So this facility is located in East Haven, and it processes about 40 million gallons of water or wastewater a day. And this actually serves a catchment area of about 200,000 people, including people from East Haven, Hamden, New Haven, and Woodbridge. And this is a 4-stage Barden-Foe treatment process with a primary and a secondary sludge incinerator. Okay, so the sludge in Connecticut is actually incinerated. A lot of other states actually will take the sludge and spread it out onto fields or use it for energy, but here in Connecticut, it's incinerated for energy. So here's a basic diagram of what the treatment facility looks like and where we got our samples. So Jordan went to the treatment facility and started getting samples daily, sludge samples from the treatment facility starting on March 19th. So if you remember back to that sort of timeline of the images of the United States when it started peaking, it was in early March when New York started really blowing up. And at that point in Connecticut, it had really moved into lower Connecticut and towards Fairfield County, and we started seeing a number of cases peaking there. In New Haven, it really hadn't taken off yet. We did start seeing some cases in hospitalizations around this time, and Jordan had arranged to collect samples, specifically primary sludge samples from the treatment facility starting on March 19th. And we collected samples daily from that point forward. Now, just to sort of explain, if you can sample wastewater at different places. You can sample it as before it comes in. So if you go up here before it actually enters the facility, so this is the effluent coming out of people's houses, coming out of schools or university buildings, whatever it may be. And that is like 99.9% water, right? So this includes bath water, dishwater, washing machines, stuff like that, as well as anything that comes through the toilets. And because of that, any virus that might be there is very dilute. And so it's hard to actually get a consistent reading on the presence or absence of coronavirus. You may sample one point and get a very high reading if you actually happen to get a piece of human waste that is hot with the virus. Otherwise, most of the time you're just going to be sampling water and not get anything. So we decided to focus on the primary sludge because at this point the virus comes into this process, sludge gets put into this gravity thickener, and a lot of the waste settles to the bottom. And so now we're looking at a solid waste percentage of about 3 to 5% is the thickness of this. So you're really concentrating the human waste material that we are interested in actually testing and getting rid of a significant portion of the water that would be there if you were taking it actually from the pipes running underneath the city. So we took the primary sludge, we mixed it up really well, and we took 2.5 mil samples of sludge. So we took them and we did that in duplicate. Each one was extracted using the RNeasy Power Soil Total RNA kit. This definitely took some optimization to find the right kits, the right process to get good recovery of RNA from the samples. And then those samples were all processed in Jordan's lab. They were then sent to me where we did the one-step RT-PCR assay, which is the primary assay to detect the genetic material of coronavirus. And we were using the CDC's qPCR assay, which included 2 primer sets that were specific for coronavirus. Is the N1 primer set and the N2 primer set, so they both target the exact same gene, within the virus genome. And then we had also one internal control, and this was the ribonuclease P. So this is a human-specific gene, and it's an internal control to make sure that our RNA extractions went well and that the PCR went well. That serves the purpose of their saying whether the sample is good enough to actually call positive or negative, or to actually quantify virus. So when we first started doing this, so like I said, we sampled from. So this study set and the data I'm sharing with you today is from March 19th to May 1st. And these two figures here are basically showing that if you look at the open circles, the open gray circles here, these represent every single point at which we took a sample. And we then quantified, this is the RP gene. And so one thing you see, if you look at the bottom, the x-axis is rep1, and for the N1 primer set. This is rep2 for the N1 and the RP primer sets. And you can see is that there's a strong correlation between rep1 and rep2, the red dots, for the N1 gene. So when rep1 was high, rep2 was high. So this is just demonstrating that using sludge could be it could serve as a very consistent source of material to accurately identify coronavirus. And at the same time, too, you see that the RP gene stays flat. So this doesn't change over time. We were consistently getting the same amount of coronavirus. Sorry, not coronavirus, human RP gene in our samples over time. And again, this over here, this second graph is basically looking at the N2 primer set. So this is the other primer set specific coronavirus. And again, we see a strong correlation here between rep1 and rep2. Again, just demonstrating the consistency of this sampling approach and our assay. So next we wanted to say, okay, well, it seems like we can detect it in the sewage sludge, and that we can reliably detect it from multiple samplings from the same sludge sample. How does the level of viral RNA in the sewage sludge, how does that map to the number of human cases that have been reported in the New Haven County? And so this figure here on the left just shows the number of new cases reported with the dates on the bottom. So you can see you've got this sort of nice bell curve happening here, where the epidemic curve, where it started out low, number of cases that were identified, gradually increasing and going down. Now, we have to keep in mind that at this early point in the epidemic, testing was a problem. So this probably did not represent all the people in the community that were actually infected. It actually is serving as a proxy for the intensity of transmission within the community. So these are not absolute values. I mean, there are absolute values in the cases that were reported, but it's not actually representative of the number of cases that were circulating within the community, because a lot of individuals were either not getting tested who are symptomatic, or, as we know, there are a number of people who are asymptomatic or pre-symptomatic that could have been shedding virus into our system. When we then compared that to our sludge data for looking at SARS-CoV-2, so we've got N1, rep1, rep1 for N2, rep2 for N1 and N2. And basically for all of them, you can clearly see that this has a very bell-shaped curve that almost matches what we see in the number of new cases. So what we did is then doing some modeling. These are smooth curves, actually in our final report, which we've has been accepted now in Nature Biotechnologies. They asked that we sort of remove the smoothing, but for purposes of this presentation, what you can do is you can smooth the data over, basically you can average them out. Any one point is an average of the 7 days prior to it. And so, and then conversely, it's also the 7 days in front of it. So it's a window of, basically you're smoothing your data out to make it look like these nice smooth graphs, as opposed to the big histograms shooting up at one point and down at another. And what we can see from this is that these, the number of new cases represented by the black line here, and the concentration or the amount of viral RNA found in the system seem to mirror each other. However, there's one slight difference, is that we start seeing a peak about 7 days earlier in our viral concentrations prior to new COVID-19 cases being reported. Right, so this was serving as a 7-day leading indicator for coronavirus cases that were being identified within our community. And not only did it serve as a leading indicator for the number of cases, it also served as a leading indicator for hospitalizations. Now, this was not as robust. As you can see, these lines kind of cross it, and it's not as clear early on with sort of these lower concentrations, but later on at the peaks, you can see that this actually ends up being about a 3-day leading indicator for hospitalizations. So by looking at the sewage sludge, and quantifying the amount of viral genetic material in the sewage sludge, we were actually able to, in a sense, get an idea of what might be coming in the future. I'm hesitant to say predict, because we didn't do a predictive model on this, but there's strong evidence to suggest that when we start seeing spikes in coronavirus RNA, that this could be an indication that there will be a following peak in human cases as well as hospitalizations. So now, can virus concentration in sludge be used to estimate the number of cases? Maybe, but the problem is we don't ever really know the true number of cases. So it's hard to do this estimate because there was so little testing early on. We didn't really know how many true cases there were. So it's hard to know whether it can actually be used to estimate the number of cases. But what I will say is that we were able to detect SARS coronavirus RNA in New Haven sludge when new cases are less than 10 per day and test positivity is less than 1%. So what I mean by test positivity, so the number of tests that are administered, the percentage of those that are actually positive. So if you test 100 people and one of them comes back, you have a test positivity of 1%. So even when we were seeing less than 10 cases being reported a day and a test positivity rate less than 1%, we were still able to detect SARS coronavirus RNA in the New Haven sludge. So, and these figures here on the right basically show, so this is basically showing at the beginning of the peak that we have a very strong correlation between the number of new coronavirus cases and viral RNA copies. So these are actually reported cases, and then this is at the tail end. So at the end of in May and June, we're still actually seeing a very strong correlation between the number of coronavirus cases and the amount of viral genetic material that we're seeing in the sewage sludge. So this is really exciting work, and this work is now it's gotten a lot of press, and there are a lot of other places that have started implementing this. I know that universities are actually doing this, and a number of universities are doing wastewater. So they're actually doing that effluent, that part that's 99% water or 99.9% water. They're testing that, and they're trying to just get an idea if there are infected individuals in a dormitory, and get a handle of what's going on at schools. I know other communities are also doing a similar thing. So the hope is that this can be expanded to include more communities across the country to start being able to get, hopefully, an indication as to where and the level of transmission is in the community. And the reason this is important is because once you have a good idea of how severe or how much transmission is occurring in the community, you can actually start implementing. You can adjust your public health policies accordingly. So if you have a 7-day leading indicator suggesting that there might be increased transmission in the community, you can say, okay, we need to shut down eating inside restaurants, or we need to go back to distance learning. Or if you start seeing a significant reduction in the amount of cases, you might be able to a couple days earlier start opening things up. So this information can really be used by public health policy individuals to determine sort of how we respond to the pandemic as a society as a whole. And so what's next for this? Well, we've continued sampling all through the summer in New Haven, and just in the last 3 weeks, we've started expanding this into other communities in Connecticut. So we are also now sampling in New London, Hartford, Stamford, Bridgeport. I think that's the majority of them. I think Norwalk as well. So we're also sampling in other places and continuing the surveillance. We've gotten funds from the state to continue doing this and expand it to other jurisdictions. So that's sort of the first half of my talk. The next half of my talk, I'm going to get into some of the stuff we were doing to actually try to improve the diagnostics in the community. And this was really led by Nathan Grubaugh. This is Yale School of Public Health, as well as Ann Wiley, who's an epidemiologist as well. And a lot of this work was done by myself and then really Chantal Vogel, who is a postdoc in Nathan's lab, and she's really been a huge part of this and really driving a lot of this. So I just want to give her her due. She's done a great job, and same with Anne. Some of the other people who've helped, obviously, is Dan with some of the modeling. Saad and Albert have been involved in this more from a administrative point of view and helping lead the projects, get samples. Albert really set up the biorepository and some of the, in order to get some of the clinical samples, he's been instrumental in doing all of that. So this is a big team effort. I want to give a shout out to Cheney Kalinich, Isabel Ott, Christina Harden, and Annie Watkins, who did a lot of, who were serving as technicians in Nathan's lab and helping doing a lot of the screening and testing in the laboratory as well. So, yep, testing is still a problem. It's a little bit different now. It's not as people not being able to get tests. We're actually having issues with having enough supplies, or the labs being overwhelmed and having delayed responses and getting test results back to people. And so we're still having a problem with testing. And so, you know, we really wanted to see, are there ways that we can improve testing here? And some of the problems are that there's a lack of a cohesive national strategy. I think everybody is aware of that. Another issue is the supply chain issues. So there's inefficient assays, and I'll get to that in a little bit. And there's over-reliance on a handful of products. So these this has led to sort of bottlenecks within the production and the distribution of certain items that are required for doing these tests. And again, I'll go into a little bit more detail on that. And then another area is that there's too many tests and not enough lab capacity. So there's regulations limiting testing to CLIA certified labs. So to be diagnosed, you have to have it done in a sort of a high complexity, CLIA certified lab. And these are sort of a roadblock to actually getting, you know, diagnostic tests in place to actually do surveillance screening. So yeah, but I'm not going to go into much detail, but it can be a problem. So what is the state of diagnostics? Well, there's over 200 different FDA emergency use authorized registered molecular and antigenic based tests for SARS coronavirus. There's LAMP assays, TMAs, next generation sequencing, RT-PCR, lateral flow assays, Western blot, and CRISPR, which is Sherlock. Even though I just named off a bunch of things, so you probably don't know what they are, the majority of these FDA-approved tests are derivations of the same RT-qPCR assay, the same one we were using for our sludge studies. However, what it is each company will submit an FDA emergency use authorization to use their specific PCR machine or their specific reagents. And this is sort of for each company to be able to maximize their profits. It doesn't help with standardizing the test across all of the labs, though. In addition to these companies all, you know, trying to get on this and have their products available and be used for testing, this has also led to a lot of profiteering off the pandemic. So there's been a lot of news articles that have discussed the price of the tests, where these inflated prices are coming from, and who's doing the price gouging. So what we wanted to do was, so our goal was to first to find places in the pipeline that could be improved to reduce costs. So what could we do to help, reduce costs and make this at least cheaper? And so here are some of the different areas along the pipeline which could be targeted. So there's the samples collection. So this is certified healthcare providers are needed to collect the samples. So from the beginning, there has been this push to be, or the primary mode of sample acquisition has been through what are called nasopharyngeal swabs. You need to have a trained healthcare provider perform this. And these are minimally invasive, but a number of people have reported them as being uncomfortable. Personally, I had one. It didn't irritate me at all, but everybody's a little bit different. So that has been a common concern with that. The fact that you have a healthcare provider having to sample this increases the cost of collection. And then you have to use a certified NP swab. It's really just a stick that they stick up your nose, but it has to be certified to be used. So there's an increased cost associated with that because not every swab is available. So the ones that are available can charge more to be used because they are specifically approved for this process. In addition, then you have to then take the swab and put the swab in a receptacle, and the receptacle then has a viral transport media. Again, these are products that have been certified to be used, and therefore, because they have been certified, they can charge more for those products. So you have an increased cost there. Then at the diagnostic testing labs, you have, like I said, they had to be tested at CLIA certified laboratories or other for-profit private laboratories who are often like Quest. And just by having them tested at those facilities, they have a lot of overhead costs associated with having to pay their buildings, the insurance, equipment, trained laboratory personnel, in addition to the profit that they're trying to recoup from doing these tests. And so that is a huge increase in the prices there. And then as far as when you get to the actual diagnostic test, some of the other places that you have increased costs are, extraction of nucleic acids. So you have to do these RNA extractions, but you can only use specific FDA approved RNA extraction kits. Again, if they're approved, then those companies can charge more. And it also leads then to shortages because there's only certain companies that can provide them and they can only produce so many kits. And so they can't always necessarily keep up with the demand. So that causes not only an increase in the price, but also a potential bottleneck in the supply chain. There's also then the cost associated with the primers and probes because we were targeting 3 genes, we're targeting the N1, N2, and the RP gene. You're having to order 3 primer probe sets, which can get expensive. Primers aren't necessarily that expensive, but probes can be. And so by doing 3 different reactions, you're actually increasing the cost associated there. And then again, because you're doing 3 different gene targets, you have to then pay for 3 times as much PCR reagents to actually run these tests. Again, only FDA approved PCR reagents can be used. So the first thing that we did is like, okay, where's a quick place we can actually try to reduce costs? And what we thought of was, can we make this three test assay and combine it into a single assay? And what we do is that's called basically making a multiplex PCR. Many of you are probably familiar with this. It was very straightforward. Just the idea was, okay, early on, can we get this into a single assay to at least reduce some of the cost at the point of diagnostics? And so what we did is we made a primer probe, a multiplex assay that had the N1 that had a FAM reporter on it on the probe, an N2 with a hex reporter, and then the internal human control, the ribonuclease P gene with a Cy5 promoter. And here we were just testing we took actually viral RNA at known concentrations, diluted it out, and just tested the sensitivity for both N1 on the left and N2 on the right to see the level of sensitivity. And by doing that, we actually showed that they could be very highly sensitive and for the detection of coronavirus. So we were consistently being able to detect around 10 copies per PCR reaction. So we wanted to validate this using some actual samples. So now what we have here is in the blue, all the blue lines on each of the 3 graphs are nasopharyngeal swabs One, green is another nasal pharyngeal swab. Saliva is another source of where the virus could be shedded for the red and the purple, and then the P is just sort of a pooled sample. And what we did is we looked at them in the form of either a FAM only, so that's what the original singleplex was. So that's with each N1, N2, and RP all had a FAM reporter, and you can see the CT values. So basically, the lower the CT value, the better detection it has of the strain. And then we did each of the new primer probe sets by themselves. So the N1 with the FAM, the N2 with the hex, and the RP with the Sci5, and compared how well that did against the FAM ones. And then we did it comparing it all with all three of them combined into a single assay. And what we find is that the level of detection is consistent especially for N1. We have a little bit of a drop in N2 when doing it in the multiplex, but not significant. We still were able to easily detect all of our samples. And then same with the RP gene, we were able to detect all of them equally well doing this. This is really just taking it sort of to the next step. So that was a very small sample size I just showed you in the last slide. This one here is actually taking 41 clinical samples that we compared the samples by the singleplex, sort of the standard assay, compared to the multiplex assay. And then we can see that there's for the most part, most samples are straight across. Every once in a while, we'll have a sample in the singleplex that has lower CTs and then higher CTs here. But they're all below the limit of detection. So you could still, even with this multiplex assay, still detect those clinical samples that were identified as positive in the singleplex. We could also detect them as positive in the multiplex. And this was consistent for N1, N2, and then RP was very consistent. Actually, you see a slight trend down for most of the RP genes where the multiplex was actually a little bit more sensitive, but that's not really that important. That's just showing that it works. These tables down here are really just showing the side-by-side comparisons, the average CT values. I just figured I'd put them up there so as you're looking at this, you can kind of compare the two. So with all this, basically, okay, so it works. The multiplex works, and it seems like that this assay can be used, and we actually do know that I think some companies have actually applied for FDA or may have even obtained FDA approval for multiplex assay. So this is just one step in the pipeline that we thought we could help. I think other people's got there before us, but at the end we're showing that okay, this is one area, and so we thought, okay, well, are there other areas we can actually improve on? And so this was really led by Ann Wiley, and this paper was just recently published in the New England Journal of Medicine. I was not part of this original study, but it's the idea that we can actually use something other than MP swabs. So now, if you remember, I said that there's costs associated with having a healthcare provider having to sample to do an invasive and uncomfortable sampling with these nasopharyngeal swabs, and that then there's a cost associated with the MP swabs themselves. And we thought, well, can we actually use saliva that somebody could just spit into a tube and use that as a source? And so when we did that, when Ann and others had done this, what they found is that when you actually took paired samples, so these are paired samples between a nasopharyngeal swab these are either inpatient individuals at Yale New Haven Health or healthcare providers that provided samples, that if you took the nasopharyngeal swabs and took saliva samples, we actually see that in some cases you have nasopharyngeal swabs that are negative, but then become positive by saliva. Conversely, we also have some saliva samples that are negative that are positive by nasopharyngeal swab. But what we see is when you kind of break it down into sort of this histogram, and this is days since COVID-19 diagnosis, that overall we actually had a higher number of samples earlier on within the first 10 days that were actually detected to be positive with saliva than nasopharyngeal. So this is suggesting that saliva not only is a comparable approach to identifying COVID-19 cases, but that it actually can serve as maybe a more sensitive method for doing this. And part of this has to do with the fact that even though people are trained to do a nasal pharyngeal swab, each person is going to be doing it a little bit differently. So there are sampling biases associated with NP swabs, whereas that mostly gets removed when you actually are collecting saliva. And to further show this, that the saliva samples compared to the NP swabs, so this is days since onset of symptoms, and one thing that we see, and I really want to draw your attention sort of down to the bottom where those nasal pharyngeal swabs were, is that there'll be samples that will be positive or negative on one day. So these are sort of longitudinally sampling. So you might sample person one day and then you'll continue sampling here, where they go one day, they're negative, but then they shoot up to be positive. And sometimes they'll be positive and shoot down to be negative and shoot back up to be positive again. And that's something you will see frequently with NP swabs. The saliva, we didn't really see that much. We saw one here went from a slight negative to a slight positive at one point in time. But for the most part, if you were positive one day, you're going to be positive the next day by saliva. So this is really demonstrating that saliva is a really good alternative to NP swabs and can reduce a lot of the costs associated with the sampling of these, the sampling of patients. So this then led us to sort of see, okay, are there further ways we can reduce costs? And that's where we came up with the idea for Saliva Direct. And this is a cheaper and more efficient means of population level screening of SARS coronavirus. And this is very simple. Basically, somebody spits into a tube, we mix it with this pretty cheap and easy to obtain protein called proteinase K. We mix it for 30 seconds, heat it for 5 minutes, and then we put it directly into the machine. So we've got no RNA extraction, no collection devices, no NP swabs, anything like that. And that was the idea going into this study. So the first thing is we wanted to see, okay, how long and how stable is virus in the saliva? And so we actually did a study where we actually took, either fresh virus and we extracted it and we looked to see, okay, can we detect the virus in a fresh sample, samples that are stored at 4 degrees, samples that are stored at room temperature, and samples that are stored at 30 degrees Celsius. And these are different concentrations of, these are basically spiked in samples. So we took negative saliva, spiked in certain specific concentrations, either 12, 25, or 50 copies of SARS coronavirus RNA, and then held them at these different temperatures. And what we found is that they are highly stable, at least for 7 days, without the need for viral transport media. So this was just by themselves. And so this is another area where we're showing that, okay, you can reduce the costs associated with having as viral transport media. You don't need to have this media to stabilize and protect the RNA when it's in saliva. The next step then was, okay, can we detect coronavirus RNA without doing an RNA extraction? Again, this is a point in the diagnostic testing step where you can actually reduce costs by eliminating the need to do RNA extractions. And so we have the samples that we had spiked in and then extracted the RNA using the traditional RNA extraction technique, and then those that we hit with proteinase K and the heat method, which I just described. And for the most part, we see that we're able to detect almost all of these samples equally well. There were 2 samples that were not detected with the Saliva Direct method, without the RNA extraction, where they were detectable with extraction. So by doing this, we are losing a little bit of sensitivity, but at the same time, we're reducing costs significantly. To further reduce costs, before we went from 3 individual PCR assays down to a single assay with 3 targets. Well, what we did is we looked back at our data and we said, okay, let's compare N1 and N2. In most infectious disease diagnostic situations, you really only have one target that you target. You target, say you wanted to see if somebody has influenza. You target one gene of influenza and one gene from the host, and that will tell you what your infection is. Well, in this assay, we had N1 and N2 both targeting the N gene of coronavirus, and we said, well, is there a need for both of those? And basically, we compared through all of the sampling that we had done and testing we had done over the duration of this pandemic, and compared the CT values, so basically how strong of a signal that you get, and we found that by and large, N1 has a stronger signal. So we said, well, do we really have a need for including N2? Is it really going to help us that much? And so what we did is we basically reduced the PCR by removing one of the primer probe sets, so further reducing costs there, and found by doing that with just a single N1 gene, so just using the N1 now, basically it's a dualplex assay as opposed to a multiplex, so it has just N1 and RP. We're still able to get pretty good detection. Again, we have a little bit of reduction in sensitivity here associated with this. So the next thing was to test reagents from multiple vendors. As I said earlier, one of the problems that we had is because there's only certain vendors that have products that are certified to be used in diagnostic testing for coronavirus, that causes that there's two things that happen there. One is that the companies that produce them, because they have a highly sought after product, just basic supply and demand, they can actually charge more for their products then. Two, that causes a supply chain bottleneck because everybody needs those reagents and all of a sudden they can't keep up with the demand. So one of the goals with this was to try to make a assay that could be adapted and used with multiple different products from multiple different vendors. So the first thing we did is we took negative saliva, we spiked in different concentrations of viral RNA, and then we tested them to see what our lower limits of detection were for using different proteinase Ks. So we used proteinase K that we bought from Thermo, from New England Biolabs, from American Bio. Basically, we didn't see any difference in the ability of any of these proteinase Ks to actually affect our ability to actually detect a coronavirus RNA. So right now, we only tested 3. The idea is that we can expand this, or other companies could easily come to us and we can work with them to include their product into our protocol so that we can increase the number of suppliers that can provide protease K to our protocol. Similarly, we also then tested our the different qPCR kits. So we have the PCR kits from NEB. So this is the NEB Luna kit, the Biorad Reliance kit, and the Thermo Fisher TAC-PATH kit. We compared all of these, and for the most part, they were all about the same, between 6 and 12 copies of viral RNA per microliter was sort of our lower limit of detection for each of these. And then we also wanted to try these on different instruments, because not every lab has the exact same RT-PCR machines. We wanted to be able to then validate that these can be applied to different machines. And so we used the Biorad CFX96, the ABI 7500 Fast, and the ABI 700 Fast DX. And for all of these, we saw very similar levels of lower limits of detection. We then wanted to compare it to the CDC assay. So this is the full CDC assay, where they are taking samples and going through the RNA extraction protocol and everything. And we then compared it to our Saliva Direct. And these were different clinical samples that we had. And again, just showing that this is doing the whole Saliva Direct pipeline. Compared to the CDC assay, we lose some sensitivity, but we significantly reduce costs. And this is actually doing it. So the TAC-PATH, so this is sort of the gold standard, the MP swab using the TAC-PATH assay. And we then compared this using saliva using the TAC-PATH. So not Saliva Direct, just saliva. So this is extracted RNA from a nasal pharyngeal swab, extracted RNA from saliva. Using the same TAC-PATH kit, we actually see they're pretty comparable, as we suggested earlier with the saliva. Then we compared the nasal pharyngeal swabs using the TAC-PATH kit, and then using saliva using the Saliva Direct. Again, if you're using a cutoff of 40 CTs, which was the new level of cutoff, we see that Saliva Direct is just as good, but you do see that our CT values do shift down. So we do, again, showing that we are losing some sensitivity in our samples. when we look at the saliva attack path versus saliva direct, so this is with RNA extractions followed by with attack path assay, and this is using saliva direct, so no RNA extractions. Again, you're seeing high lowered sensitivity, but it does very well in identifying or detecting coronavirus, and this is just showing that there was no cross-reactivity and it was all negative. These tables here are basically showing that when you look at nasal pharyngeal swabs that were positive, if you had there were 34 that were positive, and using saliva direct, we detected 32 of those 34. However, there were 3 negative nasopharyngeal swab that were detected to be positive by saliva direct. So that gave us a positive agreement rate of 94% and a negative agreement rate of about 91%. And then, yeah, I'm not going to discuss that right now. So the last thing we wanted to do is we wanted to try to make a single streamlined protocol, because each kit comes with its own protocol. Okay, how long do you do the RT step for? What temperature do you do the RT step for? What does the PCR reaction look like? We wanted to say, if we made a single universal protocol, could that be applied to all of them, just to sort of simplify things? And what we found is that in general, for most samples, you could use a sort of a universal protocol as opposed to what is the manufacturer recommended protocol, and still retain a lot of sensitivity and accuracy between using the recommended and the universal. So again, that was just more of streamlining things. And really, this is just a breakdown, a table of the products we used and the vendors, and sort of what their prices are associated with per sample. And so by doing all this, we were able to calculate that a single saliva direct test for an individual will cost between $1.29 to $4.37. And this compares to other assays that are common in this area. Rutgers and Broad Institute, they estimated it costs about $10 per test. Now, this doesn't actually account for then the actual cost that the diagnostic labs such as Quest might then charge on top of that, because oftentimes these tests get, you end up getting a bill, or insurance gets billed for $100, $125 or so. So there's still a huge problem with pricing from the diagnostic point of view. The last things I want to talk about quickly here, we started investigating, and we're trying to get this out right now, is can pooling saliva samples be an effective means to improve testing efficiency? So to increase sort of surveillance efforts when you're going back to school, is it at what point is it cost effective to pool samples, and at what point is it actually important to test every individual? And what we found is that one, when you pool samples together, you can actually detect the virus pretty well. So if you have a single individual sample versus a pool of 5, pool of 10, or a pool of 20. Now these are not using the saliva direct assay. These are all extracted RNAs, but it's just trying to show right now that can pooling be an effective approach to reducing costs associated with testing? And what we're finding is that, yep, you lose sensitivity by pooling samples. Not surprising, but what does that mean? So yeah, so this is just another figure basically showing what the CT values are versus what are undiluted 1 to 5, 1 to 10, and 1 to 20 samples. And as you can see, there is a decrease in the sensitivity of the assay. Okay, so when is a reduction in sensitivity assay okay? Well, what we're finding is that if you look at the total tests done, so if you do 10,000 tests, how many, if you have a prevalence in the community of, 0, 5, 10, 15, 20, 25, so this is percent positivity within the population, at what point does it become cost effective? And what we're finding is that around 3% here is when, if you have a percent positivity of 3% or higher, up to about 30%, pooling samples of 5 is the most cost-effective way to reduce, or cost-effective way to actually surveil the population. And however, what we notice is that down here at the lower limits, so if you have a population where we know we have very little transmission occurring at this time, you might actually be able to get away with actually pulling 10 or 20 samples at a time to even further reduce costs. So you can adjust the amount of your pooling strategy based on how prevalent coronavirus is in your community at that time. And it can be a very cost-effective way to increase surveillance efforts. So with all of that, hopefully I've introduced you to a lot of interesting and cool new work on how we are trying to optimize testing for the community, and I'm open for any questions now. Thanks.
Well, Doug, thanks a lot. You've made a lot of progress on this in a very short amount of time, and I found it to be very exciting work. So we'll go to the questions now. For the audience, you can continue typing in questions here while we're going through this. The first question is from Chris Dimka, who asks, given the assumption that the virus doesn't replicate outside the human cell, can you comment on the stability of the virus in the wastewater sludge? Yeah,
so that's a good question. So yeah, the virus is not going to replicate outside the host, out of a host cell. Viruses can't do that. But as far as in the wastewater and waste, we have yet to. It's hard to recover infectious virus from wastewater. What we imagine is that we're actually, when we detect it, we're detecting the genetic material and not actually infectious virus. It's not to say that there can't be infectious virus there. I'm sure there probably is in certain situations, but I imagine recovering infectious viruses would be very difficult. We haven't done a lot of that yet.
Can you comment on, perhaps I didn't say it, but the coronavirus RNA, what is the stability of that in the
well, I mean, so I didn't go into it at the beginning of the slide, but the virus is, is encased in an envelope, so this phospholipid bilayer that the virus acquires from the host cell when it exits the cell, and then within that, so that's it's a phospholipid bilayer, it's covered then in viral proteins, and then on the inside of that, you actually have the viral RNA, which then is basically wrapped in proteins, these nucleocapsid proteins. And this really provides some protection of the viral RNA over time. However, the infectiousness can be reduced by disturbing any one of a number of different components within the virus structure, whether it be the envelope, the structural proteins, the nucleocapsid, stuff like that. And you can still actually be able to detect the viral RNA in the sample. So viral RNA stability is equivalent to regular mRNA stability. If you leave it on a benchtop, there will be RNases that will degrade it fairly quickly. However, the virus has protections around it that help stabilize it, even though the virus is not infectious.
Okay. Next question is from Charlie Vosbrink, and he asks, have there been any programs for detecting virus of other types of viruses in wastewater that are routinely used?
So this, to be honest, I'm not 100% sure. I know that for at least for scientific purposes, yes, people have done this in the past and continue to do it in other places around the world. Whether it's actually routinely done for surveillance efforts, I do not know about that. We are specifically only looking for coronavirus right now. In theory, you could, though, do a metagenomics type of approach or have multiple different primer probe sets to different viruses to be able to do that in theory, but we're not actively doing that.
Okay, thanks. The next question is from Jason White, who asks, who notes that there have been discussion of different strains of the coronavirus around, and could this be an issue if there are multiple strains from the assays that are currently being used for coronavirus?
So so it shouldn't be an issue with the detection, our ability to detect it. However, that being said, there was a recent medRxives or bioarchives paper that described a small local outbreak where there was actually a variant in the coronavirus population that had mutated in the primer binding site for N1 that reduced its efficiency to be for the primer set to amplify up the gene. It didn't completely nullify it, but it definitely reduced its efficiency and amplification. So in theory, yes, there could be mutations in the primer probe's binding sites that could reduce its efficiency. As far as generally speaking, other mutations outside of the primer probe site will not interfere with the ability of us to detect it with this approach. If that's was the question.
Okay. Bob Mara has a question about whether the cost differential that you noted in the last part of your talk, it takes into account the labor.
Yeah, so that was the part that I was basically saying is like, so just that was really from the diagnostic and the sampling collection point of view, and not really the overhead costs and the laboratory personnel. Those were not factored into that equation. But
Okay.
and presumably it would be the same or even less because the number of hours that you would need to have a laboratory personnel actually doing this assay is reduced because you're removing the RNA extraction step. So if anything, compared to other tests, it would reduce those costs as well, but it wouldn't eliminate them.
Okay. Does anyone have any other questions? If not, then let's thank our speaker for a very interesting talk. Thanks, Doug.
All right. Thanks. All right. Bye.